All right, so good afternoon, everybody. I hope everyone's refreshed after lunch and we're ready for another set of talks this afternoon. Like Ali Ping said, I'm Emily. I'm a postdoc at Cornell right now, and I'm going to talk to you guys about a couple of studies I've worked on over the last few years, looking at this question of whether or not the host genome plays a role in determining gut microbiome composition. So many of us in this room are really interested in understanding the different factors that determine gut microbiome composition. And we know that host environment, like diet, antibiotic exposure, things like this play a large role. And when I started these studies that I'm going to talk to you about today, one of the open questions was whether or not the host genome plays a role. So a number of different phenotypes in humans are genetically encoded, things like our height, disease prevalence, things like this. So it's possible that the host genome also plays a role in determining gut microbiome composition. But as of a few years ago, we really had no idea if this were true. This was really just a hypothesis. So the open questions I want to address with the two studies I'm going to talk about today is, first, is the microbiome heritable? And when I say heritable, I don't just mean that you inherit it from your mother when you're born, but really that it's a genetically encoded trait. And second, if it is, can we identify variants in the human genome that control the abundance of the microbes living in the gut? So I'm going to talk to you about two different studies today. And the first one, I'm going to start off by talking about the study that I did in the Hutterites. So this is a study that was published a few years ago in PLOS ONE. I'm not going to be able to talk about everything in it, so if you're interested in what I'm saying, please check it out. And in the study, we aim to answer exactly these questions. Is the microbiome heritable, and what variants in the genome play a role in determining its heritability? So like I said, there are some open questions that really inspired this work. The first one was, are specific bacteria heritable? The second one is, if so, can we identify variants in the human genome that determine this bacterial abundance? And then third, can we take this out a step further, and can we actually identify what cell types in the body this host genetic variation might be acting in to determine microbiome abundance in the gut? So to do this study, I turned to a really interesting population called the Hutterites. So if you're not familiar with the Hutterites, they're kind of like the Amish. They're a religious isolate group, and they live in the western United States and Canada. So before I started grad school, I had no idea that these guys even existed, but there are tens of thousands of Hutterites in the US and in Canada. So we studied for this study a handful of Hutterite colonies, which is what they kind of call their small towns, in the southern tip of South Dakota here. And the reason the Hutterites are such an interesting population to study from a gut microbiome standpoint is that they live and eat communally. So they live in these little towns called colonies, and it's about 100 to 200 people per colony. And 3 times a day, the women in the colony go to huge industrial scale kitchens. They make a huge meal, set it out buffet style, and then everyone in the colony shows up to, that's not working, to their communal dining rooms. They take from this buffet, and they all eat together. And they do this for every single meal, every single day for their whole lives. So when we're thinking about controlling for dietary confounders in human populations, this is kind of a perfect population to study because their diet is super controlled inter-individually over time. And obviously, there's still personal preferences. You don't have to take the lima beans at the buffet line if you don't want to, but in general, things are pretty controlled. The one big difference in diet in these individuals is seasonally. So because they're an isolated population, they actually grow a lot of their own produce. They don't really use grocery stores for that kind of thing. So they have a lot more produce in summer compared to winter. So Katie mentioned this morning, one of the studies we did with this population was looking at seasonality of the microbiome, and we do actually see differences between winter and summer in this population. But for this study, we're going to kind of ignore the seasonal differences. What we're going to ask is about the host genetics. And the reason we're able to get at it in this population is because this environmental influence of diet is so controlled. So to do this study, we took samples from 127 different individuals. And first, we needed to collect microbiome data, so we asked for a stool sample. And we also needed genetic data, which we collected from these individuals as well. So using this data, the first question to ask is the microbiome heritable? Because if it's not heritable, then the rest of the study wouldn't really be anything. So typically, when people look at heritability in human populations, we look at the difference between monozygotic and dizygotic twins. So monozygotic twins have the exact same genome, whereas dizygotic twins have some genetic differences because they're not the exact same genome. So by comparing these two sets of twins, you can look at the effect of genetics in relationship to the other environmental exposures. Now, in the Hutterites, we don't have sufficient numbers of twins to actually do this kind of analysis. And with our sample size, we don't have sufficient coverage over the tree, the pedigree tree, to do an analysis that way. So we turn to this measure called chip heritability. So what chip heritability is you do a genetic association study. So you look at genetic variants all along the genome. You ask how much each of those variants explains of the phenotype, and you add up all of that variance across all of those variants. So this is a measure that's been applied to other types of phenotype data, such as cholesterol, height, things like this. And we can use it here in this study to kind of come up with a rough estimate of the maximum amount that common genetic variation can explain. So when we look at this,
the number of commonalities. I mean, I'm not quite sure what the measure is. You see, add it up. What are you adding?
You're adding up the proportion of variants that each of these genetic variants explains.
On what type?
On bacterial abundance in the gut. So what we're looking for is to identify whether or not the abundances of particular gut bacteria are heritable or genetically controlled. And so our phenotype of interest in this study is the abundance of each different common bacteria in the gut across individuals in this population. Yeah, so here I'm showing you the results of this analysis for a number of different common taxa in the gut. The purple points are the point estimate of this chip heritability, and then the error estimates are shown in these gray bars around. And so anything where the error estimate doesn't intersect 0, we think there's evidence for heritability of these bacteria. So obviously the error estimates are quite large, and we're not actually saying anything about the actual estimate of heritability. We just think that there's evidence that there is heritability of these taxa. And so as you can see from this plot, only about 8 to 10% of the bacteria that we look at show some evidence for heritability. However, this is some proportion of the microbiome, and like I mentioned, these are only common bacteria that are found in a lot of individuals in this population. So if we go back to this question, are specific bacteria heritable? Yes, it does look like certain bacteria are genetically heritable. Now what we can do is we can go forward and ask the next question of, are there variants in the human genome that are associated with the abundances of these bacteria? So to get at this question, we took a genome-wide association study approach. And this is how this works. So at a particular location in the genome, there might be a variant in the population segregating. So some individuals in this room might have a CC at this allele at this location. Other individuals might have an AA. And there are other individuals that are heterozygous and have a C and an A. And so what we're looking for is we're looking for associations between your genotype at this particular locus and the amount of bacteria in the gut. So for most variants in the genome, there probably is not an association. And so we might expect to see something like this if we stratify bacterial abundance by genotype. So here you can see, on average, there's roughly the same amount of bacteria. Doesn't matter what your genotype is at a particular location. What we are looking for is the case like this, where if we stratify bacterial abundance by genotype class, we see a difference based off of what genotype you have at a particular location in the genome. So we can go ahead and do this type of analysis for all the different variants we want to assay across the whole genome to identify in a hypothesis-free way what variants or genes in the human genome might be playing a role in determining bacterial abundance. So we do this for about 200,000 different variants in the human genome, and we do it for all of the common bacteria in our sample. And we come up with a number of hits that are genome-wide significant, but I thought I would highlight just one here for you guys today. So here, this is the entire genome-wide results for genus Akkermansia. So what I'm showing you is a Manhattan plot. So each point on this plot is a different variant in the human genome. They're ordered along the x-axis by chromosome location. And then the y-axis is the strength of the association. So the higher the point is on this plot, the more significantly it's associated with the abundance of genus Akkermansia. And what you can see here is there are 2 points here that lie above this genome-wide significance threshold. And these 2 points lie near this gene called PLD1. And when I first saw this association, it wasn't super interesting necessarily. PLD1, there's not a super lot known about it. We know that it's a molecule involved in cell-cell signaling and potentially trafficking, but there wasn't really an obvious connection to the microbiome at first. But when I dug into it a little bit more, some interesting things popped up. So from this association study, we see that Akkermansia is associated with these variants in PLD1. PLD1 is so PLD1 has been associated with body mass index in African American individuals. And why that was really interesting to me is that Akkermansia is a bacteria that's also associated with obesity phenotypes. So Akkermansia, if you have a lot of it in your gut, it tends to be protective against obesity. You tend to be leaner. So we have this interesting three-way connection between Akkermansia, a variant in the human genome that's been associated with body mass index, and this ultimate phenotype in humans of body mass index. So this is just an association across these different things. There's obviously more work to be done here, but this is an interesting direction that I think could be followed up on. So we are able to identify some variants in the human genome that are associated with bacterial abundance using this genome-wide association study methodology. And then finally, the last thing I wanted to ask in this study that I'll talk to you guys about today is going one step further and asking in what cell types are these genetic variants acting to determine bacterial abundance in the gut. We all think of the immune system as being really important to determining gut microbiome composition, and perhaps the intestinal tissues are important as well, but we also know that there are brain-gut axes interactions. And so there are a number of other tissues that might play a role in the body, and can we use host genetics to get at that? So let me walk you through the logic of this analysis. So the first thing that we know are that genome-wide association studies are typically underpowered, and especially the one I'm talking to you about today. We only have 100 individuals. That's tiny by GWAS standards. We really should have tens of thousands or hundreds of thousands. So what we do is we set these very strict significance thresholds to try to minimize false positives. But because we do that, we likely miss out on things that are real. They're just slightly below that threshold of significance. We also know that GWAS hits tend to lie in genes, or more often regulatory regions to genes. So here I'm showing you a schematic of a cell. We have a piece of DNA with a gene on it, and we have these upstream regulatory regions for that gene. We know that GWAS variants tend to lie in these regulatory regions or in the gene itself. We also know that regulatory regions tend to be cell type specific or temporally specific at some point in development. So can we put this all together? Can we look at putatively regulatory regions in a cell type specific manner to identify what tissues these variants might be acting in to determine microbiome composition in the gut? So for instance, in this fake example, we have these hits that are lying in this enhancer in a heart tissue, so we might come to the conclusion that the heart was the relevant tissue of interest. So how can we actually do this? How do we know what regions of the genome are regulatory in nature? What we can do is we can intersect our GWAS results with DNase hypersensitivity data. So if you're not familiar, DNase hypersensitivity data marks these regions of the genome that are open in particular cell types. And we think that these regions are regulatory in nature or they're genes. And so DNase hypersensitivity data is available for hundreds of different tissues because of efforts like ENCODE. And so we can simply intersect our results from our GWAS with these DNase hypersensitivity data to ask this question. So this is an analysis that was inspired by a Murano paper from 2012. And they kind of did a proof of principle for this for a couple of different diseases. So in this paper, they did a GWAS for Crohn's disease, and then they intersected it with DHS data. And let me walk you through this plot. So what you have along the y-axis is the fold enrichment of SNPs in DNase hypersensitivity sites. And along the x-axis, you have different thresholds of GWAS significance. So all of the variants in the genome are represented off to the left, and then you get more and more significant for associations with Crohn's disease as you move to the right here. And as you can see, as you move to the right for these GWAS associations, you get these immune cell types that are popping up as being significantly enriched for these GWAS variants. And this makes a lot of sense with what we know about the etiology of Crohn's disease. We know that there's a large immune component that goes into this, right? Another cute example that they have is for Qrs Duration. So Qrs Duration is on an EKG where it goes ba-doo-doot, ba-doo-doot. It's the length of the ba-doo-doot. And as you can see here, heart tissues are the ones that pop up in this analysis as being the relevant tissue type. Makes total sense, right? So we can do the same kind of analysis using the microbiome GWAS data to see what types of tissues might be relevant for this system. So I'm just going to show you a couple of examples today. I'm going to, again, show you genus Akkermansia, because as you'll guess by the end of my talk, this is one of my favorite bugs. So for genus Akkermansia, we see an enrichment in endothelial cell types. So this is kind of interesting because, as I mentioned before with Akkermansia, Akkermansia is associated with obesity phenotypes, or specifically having more Akkermansia protects you from obesity. And endothelial barrier integrity breaks down in obesity. So there's this another part of this story now with Akkermansia and this potential mechanism that could be further explored. Another example is with genus Jacala bacterium. This is a bacteria that's very common in feces, as you can imagine by its name. It lives in small intestine mucus layers, and as we see intestine and stomach tissues pop out as significant after doing permutation tests. So this makes a lot of sense with what we know about this organism and where it lives in the human body. So going back to the big picture, we can see that we can identify different cell types where this host genetic variation might be acting using methodologies like this. And obviously, this is just a first step. This is kind of just prioritizing what we think might be happening. Much more work has to be done to figure out really if these associations are real, if they replicate, and if these tissues are actually where this host genetic variation is acting. But at least this gives us a first step and some ideas of where to look. All right, so I hope I showed you a few things from the first part of this talk, which is that one, we are able to identify certain bacteria that are heritable. We are also able to identify variants in the human genome that are associated with their abundance. And we're able to identify host tissues in which that genetic variation might be acting. All right, so that was all done in the Hutterites. The next study I'm going to tell you about is very similar in its goals. And this is a paper that was published last year looking at genetic determinants of the gut microbiome, this time in twins from the UK rather than the Hutterite population. So this is work that I've done as a postdoc now. And the goal of this project was really to expand what we knew about the heritability of the gut microbiome in a much larger sample set. So the Hutterites were great to work with, and they have this great property of having this very controlled diet. However, there's only so many Hutterites. So we need to go to bigger sample sizes to be able to detect these genetic associations. So we teamed up with Tim Spector at King's College London to use their Big Twins UK resource to look at heritability of the microbiome. So if you're not familiar with Twins UK, it's a registry of twins in the UK. They've been studying these guys for a couple of decades, so there's tons of phenotype data and genotype data available for them. So this work was started by Julia Goodrich when she was a graduate student in my lab, and she is a fantastic researcher. She now just started her postdoc at the Broad. And over the course of her PhD, she accumulated over 1,000 different twin pairs from this resource, and then sequenced the stool samples from microbiome in order to do this heritability work. So in her first paper from 2014, with this large set of twins, she was able to show that the composition of the microbiome is more similar in monozygotic twins than dizygotic twins. So this points to the role of the host genome having an effect on gut microbiome composition. And importantly, when she digs down into the specific strains that might be driving this, or the specific taxa that might be driving this, it's clear that it's not all taxa in the microbiome, but only certain taxa are heritable. And so these are things like Christiansenolaceae, which is the most heritable taxa, other firmicutes. Things like Bacteroides typically are not very heritable, at least not in the UK population. So then after this 2014 study, we wanted to ask a couple more questions, digging a little bit deeper. The first thing we wanted to do was replicate some of these microbiome GWAS results that we saw in the Hutterites and in other studies. And then the second thing I'm going to tell you about today is looking at an intermediate phenotype. So can we not just look at genetic variants, but can we look at gene expression and see whether or not that correlates with bacterial abundances in the gut? So one association I'm going to talk to you about today is this association between Bifidobacteria and lactase persistence. So this was an association that was first identified by Ron Bleckman, whose study came out around the same time as my Hutterite study. And this is a really interesting association, which I'll tell you about right now, that we do replicate in the Twins UK cohort. So here I'm showing you the genome-wide association study results for the genus Bifidobacterium in these Twins UK individuals. And as you can see here, there's this peak on chromosome 2, where we have a handful of hits that are associated with a variant. This variant lies near a gene called lactase. So Bifidobacterium, I'm sure many of you guys are familiar with. This is a bacteria that's commonly found in infant guts. It has a lot of enzymes in its genome capable of degrading milk oligosaccharides, including lactose. So lactose is a sugar that is produced in mother's milk. And so in mammals, we possess this gene called lactase that breaks down lactose into simpler sugars that we can absorb. So in most mammals, you have expression of lactase when you're young and you're drinking mother's milk. So this happens in pigs, it happens in humans, any kind of mammal. However, in most mammals and humans, By the time you reach adulthood, lactase gets shut off. Most adult animals do not drink their mother's milk anymore. They're out foraging or hunting or doing whatever they do, and so you don't need to have lactase turned on anymore. However, in certain human populations, there's been selection for lactase persistence into adulthood. So in many human populations where there's been pastoralism historically, there's been selection for milk drinking in these populations. And we see that lactase expression continues on into adulthood. So that's why people like Joe Biden can put down 2 ice cream cones, and other of us, we're just looking at him with envy because we can't really do that. So let's break down this association in the twins UK and the Hutterite cohorts. So if we actually look at genotype classes for this association, we see that lactose-tolerant individuals have lower amounts of Bifidobacterium than lactose-intolerant individuals. And this is kind of counter to what I first expected to see in this study. Why would individuals that can't digest lactose themselves have more of this bacteria that can digest lactose? And let me walk you through what we think is going on. So let's say you're a person who is able to digest lactose. You drink a glass of milk. That milk passes through your stomach into your small intestine. In your small intestine is where lactase is secreted, and it breaks down the lactose. By the time that food bolus reaches your large intestine, there's no more lactose for bifidobacteria to eat, and they don't grow to high abundance. Now let's say you're a lactose-intolerant individual. You drink a glass of milk. It passes through your stomach, and the lactose passes through your small intestine because there's no lactase there to break it down. Once it reaches the large intestine, we think then that the bifidobacteria are able to use this as a carbon source. They grow to higher abundance because they're really happy, bacterially speaking. So in this model, if there's no milk in the diet, we don't actually expect to see a difference in bifidobacterial abundance between genotype cohorts. Using the Twins UK data, we couldn't actually assess this, or the Hutterite data either. Pretty much all of the Hutterite individuals drink milk, and most of the Twins UK individuals also had dietary records that included milk. However, this study came out last year in a big Dutch cohort where they had really great dietary records, and they were able to actually look at this association, and they did find this gene by environment effect. So they only found this association between lactase and bifidobacteria in the individuals that were drinking milk. So it's a pretty cool association that we've now replicated in 4 different populations. So pretty sure it's real, and I think it'll be really interesting to dig into it a little bit more, given that lactase persistence has been this huge selective force in human history. So we do see replication in at least one of these genome-wide hits for the microbiome. And then the last thing I'm going to tell you about today is, can we use gene expression to look a little bit closer at what kind of mechanisms might be happening in the gut to determine these associations. So for the GWAS I've been talking to you about, what we're really doing is we're connecting variants in the genome, or SNPs, with an ultimate phenotype. But there's a lot of steps in the middle of that, right? We think that genes or variants in the genome are affecting gene expression, which then affect protein abundance, which then maybe affect phenotype. So there's a long string of things that can happen between the genome and phenotype that really, when we're doing a GWAS, is really a black box. We don't know exactly what's happening. So would it be possible for us to shed light on part of this pathway? Can we look, for instance, at gene expression and connect that with the ultimate phenotype of bacterial abundance? And this might give us some understanding of the mechanisms of how bacterial abundance is modulated by the host. So to do this, I use the PredictScan method that was developed by people working with the GTEx data. So how this method works is using the GTEx data. If you're not familiar, this is a big cohort study where they looked at a number of different tissues from recently deceased humans. They were able to then connect that with genetic variants to identify things called eQTLs. We can then use these eQTLs to predict gene expression using other genetic data. So we can build a model using this GTEx data and then plug in other genotypes to predict gene expression in a tissue-specific manner for 40 different tissues in the human body. So what we're doing using this method is we're plugging in the Twins UK genotypes. We're using this to then predict gene expression in 40 different tissues, and then connecting this predicted gene expression back with a phenotype of interest, which in this case is bacterial abundance in the gut. So by using this, we're really hoping to do 2 things. So one is, using this method, we're hoping we'll have increased power to detect these associations. In a genome-wide association study, you're doing so many tests that it can be really hard to have power to detect things. But here, we're doing many fewer tests because we're only looking at about 20,000 genes rather than millions of variants throughout the genome. The second thing that's really cool about this test is that looking at something like predicted gene expression gives us clues into mechanism in a way that looking at just variants can't. So if we're looking at predicted gene expression, we not only know what gene is probably involved, but we also know a relevant tissue where it might be acting. So this gets us one step closer to mechanistic insights versus just a straight GWAS. So we did this on a whole bunch of different bacteria for 40 different tissues, and I'm just going to show you one example here of a result that we saw. So here I'm showing you the genome-wide results again, and here I'm highlighting for you this gene Siglec15. And this was the predicted expression of this gene in colon was associated with my favorite bacteria here, genus Akkermansia again. So Siglec15, when first looking at it, doesn't seem that interesting. It's an immune-related protein that's responsible for self-recognition. But as we dug into it a little bit more, some interesting things popped up. So in your intestine, you have villi. From these villi, you secrete mucins. This forms this barrier that protects you against invasion by bacteria, and also as food passes through your digestive system, it forms this nice barrier so that you don't get all scratched up. One of the outermost moiety of a lot of mucins is a sialic acid domain. We know that Akkermansia mycinophila has a lot of genes in its genome that can degrade sialic acid. And we also know that Siglec15 binds sialic acid. So there's this interesting interplay now between expression of Siglec15, which happens in these intestinal villi, sialic acid that's excreted by the host, and potentially the Akkermansia degrading the sialic acid. So again, not exactly sure what's going on here, but it does seem like there's a connection between these things that could be fleshed out in the future. So we are able to look at least predicted gene expression and find some associations with bacterial abundance. So from the second part of my talk, I hope you can take away two things. The first is that we are able to replicate some of these hits across populations, in particular this lactase persistence Bifidobacterium results. And we're also able to use predicted gene expressions to get a little bit closer to mechanism in some of these cases. And as a general takeaway, I hope I can show you a couple of things. One is that some proportion of the bacteria in your gut are genetically heritable. It is not the majority. It is only a small part of these bacteria that are heritable. And they're not heritable to the same degree that, for instance, height is heritable. There's moderate heritability of these bacteria. But that's really important to understand because if we're thinking about understanding how to determine microbiome composition or perhaps how to use it as a therapy, then we really need to take into account host genome for these particular bacteria if these are the ones that we're targeting. The second thing is that we are able to identify a handful of candidate genes, and some of them look quite promising, like this Bifidobacterium lactase persistence association. And then finally, some of these things seem to have apparent connections to host health. So moving forward, it'll be really important to understand this interplay between host genetics and the microbiome, moving forward to look at host health outcomes. So going back to this diagram now, we can take away this question mark. We know that to some degree, host genotype does confer our gut microbiomes. And what's really exciting is now we're acquiring more and more samples to get at this in more depth and looking at different microbiomes all over the body and seeing similar results. So if you're interested in the topic, I was only able to cover a handful of examples here today, but I've written a number of reviews covering this work and great work by other groups that are doing similar stuff, so please check them out. And I have to give a huge bit of thanks to the wonderful people I work with. I did all the Hutterite work when I was a graduate student here at Chicago, and the Twins UK projects I've been working on as a postdoc now at Cornell with Andy Clark, and I'm happy to take any questions.
Time for discussion. Please.
Good. Very nice talk. So I was wondering if I had a little bit of questions about the first point when you said is the microbiome heritable, and if not, then all of the other points are can be ignored.
Yeah.
But you showed that the rest of the points are true because there is some association of microbiome with specific SNPs. But and honestly, I didn't. Maybe it's because I don't understand the chip heritability, but I'm not sure I understand or trust because of that reason. The first answer that you had to your first question. So you said you use predictability by SNPs of specific microbes, right? This seems to be something that's highly, highly sensitive to overtraining, especially since you have so little data, 100 and something patients, and a ton of SNPs and a ton of bacteria. Can you say something about that? Like training and testing data, did you use that?
So it's not actually a classification model. I mean, it's really just a logistic regression model where we're inputting all of the different SNPs, and then we're asking the proportion of the variance explained. So it is a rough estimate, and I hope I made that abundantly clear that I was like, you know, there's huge errors around this, but they don't intersect zero, so we think that some of these things are real. So absolutely, I mean, these are super rough methods. It's not exactly heritability. It's the proportion of common genetic variants that we're assaying that we can then use to explain variability in the phenotype.
That the heritability that you showed or that you inferred in the first part by this regression, I don't know, that consists of the things that you found later in the study as well. So, for example, the Bifidobacterium lactase association and others, or is there a much more general sort of overview, like a general heritability?
Yeah, so two things. I mean, so one is that certainly for the bacteria where I detected CHIP heritability, not all of those bacteria were able to find genetic associations later. And then conversely, there are some bacteria that don't necessarily show really strong CHIP heritability or heritability in the twins UK samples where we are able to find genetic variants. So at this point, it's really a problem of incomplete power. So in both of these studies, we have sample sizes that are much smaller than what we really need to be able to detect genetic associations or heritability with really fine-tuned estimates. So my answer would be, I'm always a little bit skeptical. When I first see the CHIP heritability results, you know, these are estimates, these are a guess. What's really nice is right now we have a number of different studies that have done these methods on different populations using slightly different methods, and we all are kind of converging on similar things. So there's the Twins UK study, there's the Hutterites, there's this big Dutch cohort, there's a Canadian cohort as well, and a lot of these heritable bacteria are showing up as heritable in these different populations. So that gives a lot more confidence to these results. Not using the same method. So for instance, the Hutterites, I'm using CHIP heritability because we don't have twins. In the Twins UK, I'm able to actually use twins to estimate heritability. Whereas in the Dutch cohort, they also don't have twins. They're doing something slightly different. And in the Toronto cohort, they also do not have twins, so they're doing something slightly different as well.
Does anybody apply both methods to the same data?
Yeah, in the twins we have.
So CHIP also would align within the UK data, not just...
It does, yeah. Yeah, so the problem with CHIP heritability is that when you're doing a GWAS, you're only looking at common variants in the genome. And so you're really, it's kind of a conservative estimate of heritability. So there is a strong correlation between CHIP heritability and the estimated heritability from twins in the Twins UK, which actually the CHIP heritability tends to underestimate heritability a little bit because it can't take into account rare genetic variation. But there is a strong positive correlation there, yeah. Yeah.
The two examples you provided, Bifidobacterium and Actomyces, it appears that that host genes is just provide the appropriate substrate for the bacteria to produce. In one, it produces lactose, the other one, it produces the mucus. Is there any other example of association which is not driven by providing the substrate?
Yeah, so actually there is a really fun association that we only see in the Twins UK cohort. It has not been replicated anywhere else, so caveat, where we see association between a variant that lies in a gene that's known for cilantro's soapy, bitter taste perception. So I don't know how many of you guys hate cilantro because it tastes really bad, but there's a genetic variant in our genomes that confers when you eat cilantro, you taste this super bitter taste, and tastes soapy. That variant is actually associated with a certain bacteria as well. And so what we think is happening there is that host genetics is influencing taste perception, and it's influencing then host behavior. You know, those people aren't going and eating cilantro then, and that same variant, you know, you taste this bitter compound in a number of different foods. So likely this is changing your behavior so that you're eating slightly different things, and we think that is what is driving these differences in bacteria.
But that's eventually also substrate availability.
And availability. So
You're also changing the nutrient.
yeah. Well, and that's really the story with the lactose thing. We're not making lactose. We're eating the lactose. In some adults, they're breaking down the lactose before the bacteria can use it. Yeah.
So on this theme, what about the immune system, genes involved in immune regulation, like toll-like receptors and things like that? Are there variants that function influencing hosting in a way that either allows, or precludes certain bacteria from being?
Yeah. Absolutely. So I didn't highlight any of those results here, but we absolutely do find variants near immune genes. We see enrichment of variants near immune genes. Certainly that's the class that pops up the most in these kinds of studies, as one would expect. Absolutely, yeah. Yeah.
So you mentioned that you had about 10% of the taxa are heritable. How much does that sum in terms of their frequency, that low frequency ones or high frequency ones?
Yeah, so I should have actually been more specific. It's 10% of common bacteria that I assayed that we're looking at. So everything that I'm looking at was fairly high abundance because we need to see it to be able to do tests on it, right? So it's certainly not the majority of the microbiome. It's certainly a small percent of the microbiome, and off the top of my head, I can't give you numbers. But yeah, it is certainly not the majority of the bacteria that are there. It is a small subset that are being influenced by host genetics.
I have another small question. Just
Yeah.
about the replication. So you showed that there is a replication for the LCT Bifidobacterium story. What about the other variants? Do most of them replicate between the UK and the
No. No, and this is a problem in general across a lot of these studies. So now there's been 4 or 5 of these big GWAS, and very few of the variants replicate across studies right now. And I mean, this is a problem we're all open about, and we say it, and this is why the GWAS world requires replication. And it's not clear how much of these are a population-specific thing, or if it's just a power thing, false positive, whatever. Yeah, so I mean, the lactase bifido story replicates. That's one of the very few that we see replication across multiple studies. Yeah?
Now, in the Hutterites, obviously, okay, you control for diet. In the UK twins, that's not true.
Correct.
And yet you're finding the same bacteria in both places. So the question is, what does this say about the role of diet, if any, or is the diet among the UK twins sufficiently close to what the Hutterites are eating, so that you have kind of common diet across all of them?
So I would argue that the twins UK diet is definitely not sufficiently close to the Hutterite diet. So what I would say is that the problem with diet is it's going to increase variance, right? Everyone's going to be eating different things in the twins UK, so you're going to get this big swing in variance when you're thinking about individual bacteria taxa. So the strength of the Hutterites is that we're really reducing that variability across individuals. So I don't think it's necessarily that we would expect to see different taxa in each of these populations, but really we're just going to have more power in the Hutterites given the same sample size to detect associations. In back.
So I wanted to get to the question on the abundance. It seems that most of the bacteria cells, especially at the genus level, do variations. Is that intentional? Because I'm just wondering whether that could either mask strain-specific effects as well as take advantage of functional redundancy between strains within the same genus. So I was just wondering if there's specific strain-level gene associations that aren't consistent with species or genus level associations.
Yeah, that's a really wonderful question, and there's a couple of things there. So one is that the farthest I go down to in the Hutterite study is the genus level, and the furthest we go down to in the Twins UK study is the OTU level. So we never ever ask anything about strains, and that's because we use V4 of 16S. So we can't get down that low. But we do know that there are different strains that have different effects. I totally expect that there are going to be strains, specific strains that are heritable while others are not of the same species. And right now, we just can't see it. So I think that's definitely in the future somewhere that we want to go when we have the data capacity to do so, which we're getting to, which is exciting. The other question that you ask is about functional redundancy, and I think that's an excellent point because the fact is, our bodies probably don't really care who is there, right? Like, we're not asking names in our gut of like who's there. What we care about is that our microbiome is performing the functions that are good for us, right? And so if we were to do a GWAS of function in the microbiome, gene content, RNA content, bacterial RNA content, things like that, I think we would see some really interesting results. There's a group in China that did this with the twins data. For 250 individuals, they looked at heritability of gene content using metagenomic sequencing, and they see some interesting results. But at this point, no one has done a genome-wide association study for functional content, so we don't actually know what variants or genes might play a role. But I think that's a super interesting question. I think that's where the field is going to go for sure. Yeah.
So when you look at GWAS, and forget about the microbiome, if you look at GWAS as a predictor of something like BMI, and you do these giant meta-analyses where you pool, hundreds of thousands of people, you typically find that maybe a couple percent of the variance in BMI might be explained by all of the variants that they find. Now, that was probably an underestimate because of the false discovery correction that you were talking about. The fact that there are so many variants being tested, they have to be really stringently high in order to make it in the pool. But nonetheless, it suggests that there's a pretty small contribution of genetics to some of these phenotypes. And I'm wondering, am I thinking about this right? Do those kind of set an upper limit on what role this whole kind of scenario could play in those traits? Right? Like, so if 2% of the variance is explained by some of these genome-wide data in the population, is that the sort of maximum that could be explained through genetic effects on the microbial community on that phenotype? Or might there be some sort of other like synergistic kind of contribution?
I mean, so certainly, I mean, I think it's useful to also designate between kind of complex traits and Mendelian traits. I'm clearly a geneticist here. But so we think about the microbiome as a complex trait, right? There's probably many variants in the genome that are controlling microbial abundance in the gut. Through little tweaks here or there, right? You might have some variant that changes your pH in the gut, or some variant that lengthens your gut, or whatever. You might have a lot of these effects that are compounded that ultimately influence the microbiome. So on the one hand, yes, if we're identifying variants and they're only explaining a small percent, and I would argue that for most of the bacteria, the genetics isn't super strong anyway, so it is only explaining a little bit of this variability, then yes, that would be a problem. At the same time, there are cases, and we know of cases, where genetic variants can have large effects on the microbiome. So for example, in those OB mice that induces obesity, that has really, really large effects on the microbiome given the right diet. So it's this combination of, in a normal human population, if we're just looking at variants generally, we don't expect those to have a lot of effects, and we expect there to be a lot of them and have small effects. However, there are probably cases, and we know of a handful, where there are certain variants that have really large effects.
Okay, as a chair, I give myself the privilege to ask the last question. And so, Emily, this is a really wonderful work. And so, if we look at this from evolutionary, co-evolution perspective, and so these bacteria, heritable bacteria, you can call them intergenerational transfer, or vertically transmitted from mothers to child. So basically, this kind of bacteria may be very important to early human life. They need to be there. Evolutionarily, we need to make sure they are transferred from one generation to the next. How would you comment on this possibility?
Yeah, so that's a really great question. Actually, I'm just starting a study right now that aims to look at this. So I don't know how many of you guys are familiar with Andy Muller's paper that came out in Science last year, where they looked at co-speciation of certain bacteria along primate lineages. And we see that some bacteria do co-speciate along with primates, suggesting that they've been transferred over and over, and that they're important and they've been maintained throughout evolutionary history, and others aren't. And so I'm really interested in using his methods to get exactly at this. So do we see that these co-speciating taxa are heritable? Is there a genetic component of that? Or are they just entirely taken up by the environment? Because that's a super important question, right? Why would the genetics be influencing these particular taxa? Are they really important? Are they maintained more than other taxa? Yeah, so I don't have an answer for you yet, but in a few years, hopefully.
Thank you.
Thanks, everybody.
Thank you.